If we take a cross section of the kidney, there are two main parts, the outer cortex and the inner medulla. If we zoom in, there are millions of tiny functional units called nephrons, which go from the outer cortex down into the medulla and back out into the cortex again. These nephrons perform the major function of the kidney, which is to clear harmful substances from the body by filtering the blood. Each nephron has a glomerulus, or a tiny clump of capillaries where blood filtration starts. The stuff that gets filtered into the tubule is called the filtrate, and the rest of it leaves the glomerulus through the efferent arteriole. Interestingly, the blood that leaves these glomeruli doesn't enter the venules. Instead, the efferent arterioles divide into capillaries a second time. These paratubular capillaries then reunite, and at that point the blood enters the venules and eventually drains back into the venous system. Now, the renal tubule is a structure with several segments: the proximal convoluted tubule, the U-shaped loop of Henle with a descending and ascending limb, and the distal convoluted tubule, which winds and twists back up again, before emptying into the collecting duct, which collects the final urine. If we zoom in again on this nephron's tubule, each one's lined by brush border cells which have two surfaces. One is the apical surface, which faces the tubular lumen and is lined with microvilli, which are tiny little projections that increase the cell's surface area to help with solute reabsorption. The other is the basolateral surface, which faces the paratubular capillaries which run alongside the nephron. The urine osmolarity is the concentration of urine, and is measured in osmoles per liter, which is a measure of the solute particles that exist in a liter of urine. To help concentrate the urine, or increase its osmolarity, nephrons rely on the corticopapillary gradient, which is a concentration gradient that spans from the cortex to the papilla, which is the innermost tip of the medulla. In other words, there are a lot of solutes in the interstitium, with more solutes down at the bottom of the loop than up at the top. So as a tubule dives deeper down into the medulla, the surrounding interstitium gets more and more hypertonic relative to the lumen of the tubule. And that means that more and more water is driven out of the tubule, the deeper it goes. So you can kind of see how important the corticopapillary gradient is. It prevents us from unnecessarily losing water, kind of like a water recycling mechanism. Establishing the corticopapillary gradient takes a ton of work. Specifically, it relies on two key mechanisms. The first is urea recycling, which helps bring urea into the interstitium, and the second is countercurrent multiplication, which helps bring electrolytes into the interstitium. Let's focus on countercurrent multiplication. And let's start with blood, which is coming into the nephron with an osmolarity of 300 milliosmoles per liter. And we'll use these purple boxes to indicate the osmolarity of the blood in different sections. As it passes through the proximal tubule, there's almost no osmolarity change, so it stays at 300 milliosmoles per liter as it enters the loop of Henle. We're going to focus mostly on the loop of Henle, so let's zoom in a bit to show the ascending and descending limbs. Now, to start, let's just assume that everything is 300 milliosmoles per liter, both in the tubule and in the interstitium. The first step of countercurrent multiplication is called single effect, and it involves the ascending limb on the right side. Tubule cells along the ascending limb have sodium-potassium chloride cotransporters on the apical surface, and they shuttle one sodium into the cell down its concentration gradient, and that powers the movement of one potassium and two chlorides into the cell as well. On the basolateral surface of the tubule cell, a sodium-potassium ATPase uses ATP to pump 3 sodium ions into the interstitial fluid in exchange for letting 2 potassium ions into the cell. This helps to maintain the low sodium concentration inside the cell. Finally, both chloride and potassium move from the cell into the interstitial fluid as well, passively through their own channels down their concentration gradients. So essentially here you have solutes being transported out into the interstitial space. But since we're in the ascending limb, it's impermeable to water. So the tubular fluid becomes more dilute as solutes are continuously transported out. So we go from 300 to 200 milliosmoles per liter. Now, the volume in the tubular fluid is a lot lower than the volume in the interstitium. But if this process happens over and over, then the interstitial fluid will become more concentrated, and eventually rise from 300 to 400 milliosmoles per liter. In the descending limb, the tubular fluid boxes have an osmolarity of 300 milliosmoles per liter. But unlike the ascending limb, the descending limb is permeable to water and solutes. So that means that the tubule will equilibrate with the interstitium. By the process of osmosis, water will move from the tubule to the interstitium, and solutes will diffuse in the other direction, from the interstitium into the tubule. Once equilibrium is reached, osmolarity in the descending limb will go from 300 to 400 milliosmoles per liter. The second step of countercurrent multiplication is called flow of fluid, and it involves new fluid entering the kidney and going through the nephrons. To illustrate this, let's push two new boxes of 300 milliosmoles per liter into the descending limb. And this shifts everything over, and bumps two boxes out of the ascending limb into the distal convoluted tubule. Now that that's happened, remember that there's also the single effect in the ascending limb. So sodium is reabsorbed from the tubular fluid boxes and moved into the interstitial fluid. In the two boxes near the bottom of the ascending limb, the osmolarity will drop from 400 to 300 milliosmoles per liter, and likewise the 200 blocks will drop to 150. Then all these interstitial osmolarities will transfer to the descending limb. The reason these top guys at 150 milliosmoles per liter don't drop all the way to 100 milliosmoles per liter is that as the ion concentration falls, it's harder and harder for those sodium-potassium chloride cotransporters to find and move ions, because there literally aren't as many of them around. Doing another flow of fluid step gets us to something that starts to look like a gradient. And if we continue to repeat this cycle over and over again, the corticopapillary gradient basically takes shape, with an eventual concentration of 1,200 milliosmoles per liter at the inner medulla, where there's a very high concentration of sodium ions in very little water, and 300 milliosmoles per liter in the outer cortex, where the concentration of sodium and water is similar to the blood. Now, it's important to keep in mind that there's an additional process called countercurrent exchange that happens in the peritubular capillaries that travel alongside the descending and ascending tubule. These peritubular capillaries are permeable to both water and solutes, and blood has an osmolarity of 300 milliosmoles per liter. So along the descending limb of the tubule, water just diffuses out of the blood into the interstitial fluid, while solutes diffuse in. And if this blood just carried away the solutes, it'd destroy the corticopapillary gradient. To counteract this, though, the peritubular capillaries also ascend alongside the ascending loop. Since the corticopapillary gradient decreases as we go up the ascending limb, the extra solute in the blood diffuses right back out of the vessel and into the interstitium, and water diffuses back into the vessel. So as the blood exits the loop of Henle, it has an osmolarity of 325 milliosmoles per liter, which is only slightly higher than when it entered. In other words, water is secreted into the tubule, but then reabsorbed and returned to the systemic circulation. And solutes are reabsorbed into the circulation, but then secreted back into the interstitium. And from that, only a little bit of solute that helps form the corticopapillary gradient is lost. This helps maintain that gradient that took a lot of energy to create. The amount of solute lost this way depends on the rate of blood flow. Normally, the blood flows slowly through the peritubular capillaries, and that allows time for passive diffusion to maintain the gradient. If the blood flow increases, though, then there's less time for the diffusion of solutes out of the peritubular capillaries at the ascending limb, and more solutes will be carried back into the systemic circulation. Alright, as a quick recap, countercurrent multiplication is the continual repetition of a two-step process that gradually increases the corticopapillary gradient. For the first part, single effect, in the ascending limb, electrolytes move from the urine to the interstitium, while it's impermeable to water, so the urine becomes more dilute. In the descending limb, both electrolytes and water can diffuse across, so it comes to equilibrium with the interstitium. Then the second part is flow of fluid, which pushes fluid of different concentrations through, which establishes the corticopapillary gradient where there's an increasing osmolarity in the medulla, which ultimately helps more and more water get reabsorbed from the tubules.